Hello, everyone. My name is Dazhi, and a PhD student at Essen University, working with Dr. Yuan Dickerson. So today, I'm really glad to share you some one of our recent works about the learning brain organizations from different imaging data sets. So as we all know that the parcellation subdivide the brain into distinct regions based on different properties, such as the early work of the Brodmann areas, which mainly based on the anatomical information. Later on with the fMRIs, many parcellations based on the functional connectivity data has been developed, which aim to parcellate the brain into homogeneous functional region. And brain parcellations, really plays a key role to help us to understand the brain organization. And it has many practical usage, such as in the dimensionality reduction or define region of interest RIs for subsequent analysis. But when we use them to map a certain function to a brain region, then we find the anatomical parcellations cannot reflect the true functional boundaries. So here I'm taking a cerebellum as an example. So if we look at this brain activities during different tasks, the activation maps of these tasks actually falls into very different places, and none of them are actually aligned with the anatomical boundaries at all. So this really motivate us toward the use of the functional parcellations, and many works has been proposed based on resting state functional connectivity data or the task-based fMRI. So this is quite nice. And if we quantify how much of these proposed parcellations correspond to the the functional task boundaries using an evaluation metric called DCBC, then we see a most recent task-based parcellation MDDB10 show a better performance. So the idea of this evaluation assumes that if there's a true boundary, then the functional response of the two voxels that are assigned to the same parcel should have higher correlation than the two voxel pairs with the same distance that are assigned to the different parcel. So if there's no boundaries at all or random parcellation, then the coefficient should be 0. So we use this evaluation to evaluate these parcellations, and we see the anatomical parcellations perform just above chance, but the functional parcellations perform quite well, and our task-based parcellation slightly outperform the resting state, which is quite nice. But from a practical usage, we are not really happy about these parcellations, because when we look at this parcellation, we see that there are still some areas that are highly uncertainty or noisy. So for example, the foot motor regions that are believed on top of the cerebellum are pretty noisy. That is because there was no foot motor tasks that included or involved in the data set that this parcellation was trained on. So if we really want these areas become visible, then we probably need another data set that has this knowledge. So this really limits the utility of these parcellations, because the most of existing parcellations are developed based on single fMRI studies. But the learning of a comprehensive brain organizations usually need a large enough battery across many functional domains with enough number of subjects. So, but this is currently not available in current neuroimaging studies. So instead, we have right now is this increasing number of different types of fMRI dataset. So if we represent them in a square, which the horizontal is the number of subjects and the horizontal, the vertical is the amount of data per subject, then we see 3 base types. For example, one type of the dataset could have a lot of subjects, but relatively few amount of data per subject, such as HCP resting data, or we could have a lot of task measures per subject. But these types of dataset are usually smaller size in terms of the number of subjects, which we call this type as a deep phenotyping dataset. And you can actually build brain parcellation based on this type. And also there are some other dataset comes available somewhere in between. So we wonder, can we actually take advantage of different data set and fuse this information together into a single model and actually build brain parcellation from it? So this is our initial motivation of this project. To this end, we propose a hierarchical Bayesian framework. So the central quantity as well as the outcome of the framework is this individual parcellations or probabilistic or soft parcellation that links each part of the model. The top level part is what we call the spatial arrangement model. So this provides a group probability measure of how likely a certain parcel assignment of the brain locations is across the study's populations. So for example, you can think for a single brain location I, then we can represent the parcellation of a specific subject S as a multinomial random variable US across all K regions to determine the probability of this location I belongs to which parcel K. We then can concatenate these vectors as a big matrix to get a group probability map of all brain locations. And this big U given some model parameters. The lower level part is what we call the data emission model, which provides or emits the knowledge of the data set. So these emission models are modeling each fMRI data set separately and calculate the likelihood of a certain subject given the brain organization of that individual. So usually this is a K-mixture model. And a nice thing of such a structure is that we can handle the within subject variability. For example, if we have multiple imaging sessions in a single fMRI study, like the dataset B here, instead of simply concatenate or average the data over the two sessions, we model them separately and really capture the measurement noise. During the learning, the emission model calculates each of these subject-specific data likelihood and pass them up to the central arrangement model. And now the job of the arrangement model for every subject is to calculate or approximate the posterior probability of P of U given the data likelihood and the parameters. I should mention here, in many cases, there has no closed form solution, so we need to come up with a proposal distribution. And this posterior expectation denoted in this square and angle bracket are not only our best guess of the individual parcellation, but also quantify the uncertainty across the populations. This is very important. And then this expectation passed down to the emission model to calculate the data likelihood for the next iteration. And then we learn this full model through multiple EM process until convergence. So for the learning, what we want is we eventually want to maximize the log evidence given the overall model parameters. But the computation is intractable. So given the variational inference, we can turn to compute the evidence lower bound as our new objective function. And the big advantage of this split structure is that the objective function and the overall parameters now can break down into like their belonging model, and can be the model parameters can be learned separately, making our model computationally very efficient. So now let me show you some interesting results. So first, we applied our framework to learn a fusion parcellation from 6 functional task dataset. So for comparison, we also trained the parcellations using each of the single dataset standalone. So what I'm showing you here are the trained group parcellation. So we can see the maps trained by this single dataset are all looks differently as they are having different, of course, they are having different task measures. But the fusion map show the combined strengths across this dataset and not inheriting their weakness. For example, the foot motor regions are now becomes visible, which we can never observe in the data in the maps trained by this middle 4 dataset standalone, because they don't have such foot motor task involved. And this knowledge is actually learned from the first and the last dataset that has this related task, making our fusion maps has the combined strengths. And of course, the evaluation result on an independent test set also show the fusion map significantly outperform the maps trained by the single dataset standalone. Another benefit of using our framework is that we can improve the individual parcellations. So previous studies usually use group map for individual RI definition, but this is not very convincing because it cannot actually reflect the individual organizations because of the individual variability. So if you look at this individual map, they do preserve some common structures, but the functional region are all looks differently in terms of the size or the shape of each individual, and the quality of these individual maps are also suffered from limited data. So our framework actually bridged this limitation by optimally integrate the group probability map to the limited individual data. So in this experiment, we trained the individual parcellation using different amount of data with or without combined group map. As we can see, the individual map based on the only 10 minutes of training data is generally noisy, and the evaluation result also show a lower performance. But meanwhile, if we plot the group map as a comparison, we see the individual map learned from 10 minutes data is even worse than this group parcellation, which means we can simply use this group map rather than waste time to scan these 10 minutes of individual data. And for sure, the performance of the individual parcellations improves continuously to beat this group map when we use more data and reach the peak at 160 minutes. But as a trade-off, we need to scan more imaging data for that individual. So in our case, you actually need about 60 minutes individual data to beat or just like even as good as the group map that you can download for free from the internet, and need about at least 110 minutes scanning data to substantially better than the group map. But as a contrast, by using our framework, you will actually get a pretty good reconstruction, even using 10 minutes of individual data, as can be seen in this red line, which already substantially better than the group map at 10 minutes, and roughly as good as your trained model on 100 minutes data standalone. So this is a really big improvement. Okay, to sum up, the lack of a large enough homogeneous task dataset really limits the study of brain organization. So to address this problem, we built a hierarchical Bayesian framework to optimally learn brain parcellation from different fMRI datasets. And then we found the resulting individual and the group parcellations are all like improved. So for more computational details and other results of this project, please check out this paper. And thanks all the collaborators and the grant support. Thank you. Thanks for your attention.